Welcome back guys, welcome to another video from Penglam Kwak. This is the fifth lecture series of plant anatomy. Here I'm going to talk about in details each and every parts of simple pitch and border pitch and the difference between simple pitch and border pitch. Before I dive to the main topic, first you need to have a clear concept of cell wall and its composition. I have been talking about the various type of cell wall composition in plant anatomy video lecture series 3. I recommend you to first watch that video and after that you come back to this video. Let's dive to the main topic. This is a picture which represents a plant cell, and plant cell is made up of three components, that is medial lamella, primary cell wall and secondary cell wall. The medial lamella, it will be white in color, primary wall it will be blue in color, secondary wall it will be brown or olive in color. Here if you look the plant cell wall, let's represent this cell 1 and this one it will represent cell 2. Pitch means they are the characteristic depression. This here is a depression, that means a hole or no secondary cell wall is deposited here. And this depression it acts as a channel for transport of waters and mineral between two adjacent cells between 1 and 2. This depression is known as pitch. There are two main types of pitch, simple pitch and border pitch. In border pitch, the pitch will be having some border or some secondary wall will be surrounding the pitch, but in case of simple pitch, only a pitch will be present, no border or no secondary wall will be surrounding the pitch. Out of two types of pitch, that is simple pitch and border pitch, first let's discuss simple pitch. Secondary wall deposition is not uniform all over the primary wall. If you look at this picture, this brown or olive color represents the secondary wall. It is not uniform or equally distributed here, and instead of equally distributed, pits are present. That means secondary wall is absent. Certain small area of variable shape remain thin. This portion remain thin, and it is called a simple pit. This simple pit will contain plasmodesmata. And simple pit consists of pit cavity together with the primary wall, which close the pit. Pit can develop over the primary pit field or the primary wall devoid of pit fields. Simple pits do not contain border, and thus only a pit should be present because secondary wall is not present within the medial lamella and the primary lamella. This picture represents a simple pitch on vessel wall. Vessels can be of two types, xylem vessels and phloem vessels. Within the xylem or phloem vessels, small amount of holes were present. They are called simple pitch. B, that is sectional view of simple pitch. If you see here, this one will be the secondary wall. Next to the secondary wall, primary wall will be present, and next to the primary wall, medial lamella will be present, and next to that one, another primary wall will be present, and another secondary wall will be present. Let's assume that this portion represents cell A and this portion represents another cells B. Within these cells A and B, plasmodesmata were present because through plasmodesmata the protoplasm of living cells are interconnected, and I have already discussed the function of plasmodesmata on plant anatomy lecture 3. Generally, its pitch has complementary pitch exactly opposite to it on the wall of the neighboring cells. Such pit form a morphological and functional unit called pit pair. So when we talk about pit pair, here is another pit for cell A, this portion, and here is another pit for cell B. So these two combine together from cell A and from cell B. The pits combined together, it's form a pit pair. If you see this picture, let's assume this side is cell B and this side is cell A, and these are the cell wall, but within this cell wall Here you will not find no more deposition of secondary cell wall. This will be the secondary cell wall. Next to that one, primary cell wall, and in the middle it will be middle lamella for cell B, and for cell A, primary cell wall, and after that one, secondary cell wall, and here middle lamella will be present. And the cavity formed by the break in the secondary cell wall is called pit cavity. This cavity is formed due to the activities of absence of secondary cell wall. Because of that one, it is called pit cavity. And the primary cell wall and middle lamella that separate the two pit cavities of a pit pair is called pit membrane or closing membrane. Here, primary cell wall and middle lamella will be present, and which separate the pit, it is called pit membrane or closing membrane. And the pit opening is called pit aperture. This opening, it is called pit aperture, or sometimes it is also known as pit cavities. Ramified pits. In cells with thick wall, the cell wall is too thick, the simple pits may break into two or more cavities and look like the branch canals. This type of pits, which contain many branch pits, they were called ramified or ramiform pits, and mostly it is present in case of simple pits. The second pit, that is border pits, and this border pit is much more complicated than simple pits. If you see this diagram, here the secondary cell wall will overarch or overhang, will overproduce from both sides over the pit cavity. This is the pit cavity. Over the pit cavity, this black portion is called a torus, and they contain the pit membrane. Here primary and middle lamella will be present, and they will leave a narrow pore at the center. This one, they will leave a narrow pore at the center. Because of this one, it is called border pits, and they contain a border from here, here, border pits. In simple pit, no such development were present, and the empty region covered by the overarching of the secondary wall is called the pit chamber. This empty region covered by the overarching overcovering of the secondary wall by the secondary wall is called pit chamber. These elevated overarched secondary walls open to the cell lumen by the pit aperture. This is called pit aperture because of secondary walls. Pit aperture were formed, and as the secondary wall is usually very thick, a canal is formed in between the pit chamber, and cell lumen called pit canal. Pit canal will be formed here. Different types of substances can flow from one cell to another cells, and the pit canal opens to the cell lumen by the inner aperture. By inner aperture, some substances can pass, and by the outer aperture, from outer aperture. And in front view, the border pits exhibit two circles, the pit cavity forming a border around the pit aperture. In general, each pit remain in pairs. Pairs mean two. One pair it will contain two persons or two substances. As complementary pits opposite to each other occur in the neighboring walls. Let's assume this is cell A and this is cell B. And here this will be the cell wall, and here this will be the middle lamella or some portion of primary wall, and this will be the secondary wall, and this will be the secondary wall. And here we have a pit, cell A pit and cell B pit. These two combined together form a pair. A pair of simple pit form a simple pit pair. Half border pits. When a border pit is opposed by a simple pit, this is a border pit surrounded by secondary wall opposite to simple pit. This type of pits are called half border pits. Blind pit. If a pit has not opposing pit, occurs if pits open into intercellular space. If there is one pit on one side but another cells does not open a pit, the type of pit will be called blind pit. Unilateral compound pitting. Uni mean one. A large pit let's assume this is one pit very large and is opposed by two or more small pits. If there is two or small pits were present opposite to large pit, the types of pit will be called unilateral compound pitting. Let's talk about Vestured pits. Medial lamella will be present, after that next to the medial lamella primary wall, and next to the primary wall secondary wall. In this case also primary wall and secondary wall. In some dicot plants, pits with minute outgrowth. These are minute outgrowths from secondary walls surface of a pit chamber. These are called Vestured pits or sieve pits. This pit appear as a porous or net-like in top view. And this type is considered as advanced type as it is found in phylogenetically more advanced xylem. Phylogenetically means a way that relates to evolutionary developments and diversifications of species or groups of organisms. These Vestured pits are of different types. It can be filamentous, just like a finger-like projection, or coralloid, etc. All the Vestured pits are obviously border pits. That means all Vestured pits will be border pits, but all border pits are not Vestured pits. Another important term, that is Torus. The Torus, it is present in border pit, but it is absent in simple pits. The pit membrane become thick. This slide in its center portion and is termed the Torus. This is the pit membrane, and the center portion will become more thicker. And this size, this black in color, the diameter or the size, it will be much more bigger than this pit aperture. So it is called Torus. Another term, that is Margo. Margo, it is present in case of tracheates, that is xylem vessels of many conifer, big plants, the Gymnosperm. The pit membrane, the membrane which surround the Torus, is called Margo, and this Margo, it is porous, that is hole are present. And this pit membrane, the Margo, is flexible, it has some elasticity. This represent the porous of membrane and Torus of pinus. Pit aperture open and pit aperture closed by Torus. Torus is very important. Under certain circumstances, and the Margo move toward one or the other pit aperture, closing the same with the Torus, as in figure. This is the Margo and this is the Torus. So this Torus is surrounded by the Margo, so it can move sideways, this side and another sides. In this condition, water does not move through the pit and is called aspirate. So when the Torus and the Margo move on one side and block the pit aperture, so that water will not move or will not pass, and that time it is called aspirate. That means it block the passage of water. Then when the Margos and the Torus together act as a valve, that means it allows the passage of water only one direction, but here in both direction water can move, but here only in one direction. So pit aperture open here, pit aperture closed by Torus. At that moment it is called aspirate. Aspirate pit. If the membrane flexibility is lost, this membrane flexibility, that means it cannot pass from left hand side to right hand side. The Torus block the pit and the aperture permanently. The pit is called aspirate pit. It will not allow anymore the passage of waters or different substances from one cell to another cell. And this type of pit is found in most of the tracheates of the late wood, that is mostly the dead cells, and all of them in the hardwood. Another term of hardwood, that is duramen. Mostly these hardwood, they are dead, and it is found in the central portion of the wood. Comparisons of pits of conifers and angiosperm. Both angiosperm and gymnosperm possess border pits. However, they show considerable variation in their ultrastructure. That means if you watch or observe under the microscope, the variations are mainly shown in the ultrastructure of the pit membrane. The pit membrane is totally different. In angiosperm, the pit membrane is homogeneous. That means you will not be able to define which one is the Torus or which one is the Margo. But in case of gymnosperm, the pit membrane is heterogeneous, different composition. Structure of the border pits in the tracheates of the gymnosperm is more elaborate. That means here Torus is present and Margo is present, and you can easily differentiate the parts. Difference between simple pits and border pits. Simple pits occur mainly in parenchyma cells and are rarely in sclerenchyma cells. These parenchyma cells in this photosynthesis, and they can store starch or glucose. And sclerenchyma cells, they are strong and mostly for support of the plant. In border pits occur in sclerenchyma cells only, absent in parenchyma cells. Simple pits found in medullary ray extraxillary fiber component cells and in tracheids of some angiosperm in flowering plant. Abundantly found in vessels of angiosperm. Vessels mean xylem phloem, that is, and this angiosperm in flowering plants, and tracheids of gymnosperm and ferns. Gymnosperm, those big trees. The seeds will be naked. Pit cavity remain in the same diameter throughout. The pit cavity remain the same diameter from here to here. Size and shape of pit cavity varies. If you see here, the pit cavity here it is smaller, and in the middle it become more bigger. At the same time, another side it become more narrow again. Closing membranes of pit remain in the same diameter throughout. The membrane, the closing membrane, it remains same diameter throughout. Closing membrane of pit varies in their diameter through development. Here it is small, and after that one it is big and small. Simple organization of pit membrane. The pit membrane were simple organization. Pit membrane were complex with complex organization. Here the pit membranes, it contain Torus, and after that one Margos will be present. Pit border absent. Here there is no pit border. No secondary wall depositions take place within the pit aperture. But here secondary wall deposition take place in the border pits. Because of that one, it is called border pits. Simple pits occur in a thin wall are shallow, whereas in thick wall, the pit cavity may have the form of a canal passing from the lumen of the cell toward a closing or common pit membrane. In the case of thick secondary wall, this is a thick secondary wall. In border pits, the border divide the cavity into two parts. Number one, the space between the closing membrane. This is the closing membrane. All this is a closing membrane, and the pit aperture. This small hole, they call it the pit aperture. This empty space they will call pit chamber. The canal, all the way leading from this pit chamber to the lumen of the cell is called pit canal. Pit canal allowed the passage of nutrients, microbodies, etc. from one cell to another cells. Pit membrane homogeneous, which means made up of same kind. The membranes or the membrane, they are made up of same kind. But in border pits, the pit membrane homogeneous in angiosperm flowering plant and heterogeneous, that is different kind, in gymnosperm. Gymnosperm are those plants which have the seeds are naked. Torus is absent. This black in color, it is absent in simple pits, and torus is present in border pits. Torus is thickening of the pit membrane. If this is the pit membrane, it will thicken at the center of the pit membrane, which acts as a valve. Valve means or a device or a substance for controlling the passage of fluid or air through a pipe, duct, etc., especially in automatic device allowing movement in one direction only. Valve light openings and closing mechanism absent. It is absent since they do not have torus. Torus and Margo. Margo means a substance which cover the torus. This together they'll work and with pit aperture, X light valves, and which regulate the opening and closing of the pits. If this torus and the Margo were here in the left hand side, and if it's closed, then it can regulate. It will not allow the passage, the flow of nutrients or microbodies from one cell to another cell. This torus also they can move to the right hand side, and if it's after that one, it can block the passage of water, nutrients, microbodies from one cell to another cells. Plasmodesmata connection are seen in pit membrane. In simple pit, plasmodesmata will be present, and this plasmodesmata, they are the product of endoplasmic reticulum. but in border pits, plasmodesmata connections are absent. I hope that's helpful.